Hi, my name is Isabelle Cullen. I am a student in the Smear Lab in the Institute of Neuroscience, and today I'm here to talk to you about my summer research project through the Hours program. So in the Smear Lab, we primarily investigate olfactory navigation, which is the ability for an animal to locate an odor source using its sense of olfaction. So this is very important for being able to identify predators, food sources, and danger. So how we test this is using a freely moving paradigm where a mouse is placed in a box as seen in figure A. Mouse is placed in the box and their task is to go to the side of the box that has the higher odor concentration, a pinene, which is just a neutral odorant for the mice. So we're able to analyze the olfactory navigation through a small device implanted into their nose called a thermistor, but also video data that denotes their body position. So through our analysis, we found that at specific points in the task, animals perform different body movements. So sometimes they pause, sometimes they run to the left, sometimes they run to the right, as seen in figure B. And in figure C, you can see we have each color in this graph actually represents a different behavioral motif. So as you can see at the beginning of the task, they perform more of certain movements versus at the end of the task where we see a different movement. And these are just broken up by right versus left trials where the odor concentration was higher, and also broken down by time that it took the mouse to locate the odor source. So that leads us to question, what is going on in the neural system that is informing these movements? So we're curious if glutamate could be the main informant. So how we tested this was through a viral injection procedure, and that's what my main portion of the project was last summer. I helped figure out the virus injection procedure within the olfactory bulb. So what this actually is that we take a piece of DNA and insert it into the olfactory bulb to make glutamate receptors create green fluorescent proteins, so that when a glutamate binds to the receptor, it then fluoresces and we can see where, and we're having patterns of activation with glutamate. So you can see an example of this in figure E in vivo. So this is an image of the olfactory bulb, and each little circle that you see is an individual glomeruli, and it's lighting up because there's a lot of glutamate binding to that area. So you can see examples of what it looks like when different odorants bind, so methyltiglate to hexanone and ethylbutyrate, and the changes in fluorescence that we see with that. So how we actually do this is we make a small incision above the scalp and drill into the olfactory bulb so that we can inject the virus into a small area called the mitral cell layer. So in figure G, you can see this is a histology image of the olfactory bulb. So there are 4 different layers. There's the glomerular layer, the periglomerular and tufted cell layer, which is in figure in 2. And then the layer that we're trying to get at is number 3, which wraps around the inner neurons and is probably responsible for the transduction from odorants into higher movements. So that's what we're most curious about. And we're still currently developing this technique in the lab. So once we've been able to successfully implement this in a number of mice, we check the expression of iGluSnFR in the olfactory bulb using 2-photon imaging, which you can see in figure H. And what this looks like in vivo is denoted in figure I. So those little green circles that you see are individual glomeruli lighting up in response to just the activation that the 2-photon microscopy actually provides. So once we've done that and checked that it's actually working, our ultimate plan is to be able to perform the freely moving olfactory paradigm while recording data from the olfactory bulb and seeing how glutamate responds at different movements. So if during the pause we see more or less glutamate, and repeating the same paradigm and seeing how glutamate actually informs these movements. With that, I'd like to acknowledge and thank my lab for all of their help in developing this poster and their mentorship throughout this, especially to Dr. Matt Smear and Teresa Finley, who have been phenomenally helpful in creating this project.